Welcome once again to the BP Endure seminar series. And today we have the pleasure of listening to Dr. Damon Clark, who is Associate Professor at the Department of Molecular, Cellular, and Developmental Biology and Department of Physics at Yale University. Eh, it's interesting when we always want to have our students understand how science can be so transdisciplinary. And neuroscience is one of the examples. And Dr. Clark is an example of how you can connect two disciplines into one in answering very important questions. And since he was in his academic process, he became trained as a physicist, first at Princeton with his bachelor's degree, followed by a PhD in physics at Harvard. And there he studied how and specifically being I would call him a biophysicist then in principle, trying to understand, how the small nervous system in C. elegans was a different, the worm, no? And how it encodes temperature preference behaviors. And that looks a lot like Daniel Colon Ramos' work that he studies as well, how, C. elegans interacts with its environment and the genetic component of learning how to interact with your environment in such a wonderful model. However, Dr. Clark decided that no, C. elegans is not very the one that I really want to focus. So he decided to change to a very important and very attractive model, which is the Drosophila model, the fruit fly. And then when he was doing, that was when he's doing his postdoctoral work at Stanford, where he then embarked in a journey to understand the visual system of this wonderful model in neuroscience. And now that he's, a professor and committed to his students and committed to his research, he's now much more interested in how small networks of neurons perform sophisticated computations that guided behavior. And this is very important because, again, you see the transdisciplinary from the point of having computational biology, physics, behavior, neuroscience. So this will be a wonderful opportunity for all of us to see how we can interact all these disciplines in a wonderful story of science. So thank you, Dr. Clark, for being with us today, and we look forward to your presentation. And before I finish, I would like to say that after Dr. Clark's presentation, Dr. Charles Greer and him and Dr. Clark will talk a little bit about what Yale has to offer for training programs, not only at the graduate level, but in other venues. So thank you, Damon, for being here.
Thank you so much for inviting me. It's a pleasure to be here, and I'm really looking forward to interacting with you guys. So I guess one thing I wanted to make sure of is the goal of my talk is to get you as excited about this sort of research as I am, and so I really want your questions. If something's not clear or you're interested in hearing more about something, just by all means unmute yourself, interrupt. Like make sure that this is an interactive presentation. And I guess if you're shy about speaking up, you can definitely just type something into the chat window, and I have that open on my computer, so I'll keep an eye on it while I'm giving the presentation. So I'm interested in trying to understand how networks of neurons perform basic computations. And so really what this comes down to for me is understanding how visual processing happens. And in particular, I'm interested in motion perception. So I have a couple of videos just to start off here. So this is something I found on the GoPro website. So this is a bicyclist, a mountain biker, who is navigating along this pretty treacherous path on a mountain and is using visual cues and especially motion cues in order to avoid death and do all of these acrobatics. And so for navigation, motion is just an incredibly important visual cue. And this is true for sort of crazy humans, but it's also true in many for most other animals. So this is a large lizard that lives in sort of a horrifying valley here. And as the snake approaches, the lizard detects motion. The snakes are all detecting motion too. So you see all these snakes kind of slithering out of the rocks to chase down this lizard, and they're all detecting motion. The lizard is using motion cues in order to avoid the predators. The predators are using motion cues to chase the prey, and then strike at it. This has a happy ending, so the lizard actually gets away here. I'll wait till we see the happy ending here. So the lizard makes it to safety. But these motion cues are critical both for the navigation and also for sort of identifying other animals in the environment and that kind of thing. And so my lab wants to ask a few fundamental questions about how the eye works to estimate motion. So at sort of one level, we want to understand mathematically what's going on. So how, if you were going to write something down on a chalkboard to describe like what your eye is doing when it detects motion, what would that be? What would the equations be? At kind of a lower level, we really want to understand what the mechanisms are that generate those equations. So how do the biophysics of these cells work? How do they interact with each other via synapses? How can you basically, how does it work at like a cellular level? And then at kind of one level up, we want to understand why evolution has arrived at a particular algorithm to solve this problem. And so we spend time thinking about how the eye works with natural inputs and that kind of thing. And one of the exciting things I think in this research is that at these two levels, sort of at the algorithm level, so like the mathematics, and at the kind of understanding how this relates to like environmental inputs and that kind of thing, there are actually a lot of similarities between very different animals. And so one of the things we'll talk about a lot today is the similarities between humans and fruit flies. So I think, in neuroscience, we're all interested in trying to understand this organ, right? So this is a human brain. It's rather large. It's very complicated. It definitely creates motion signals. So it definitely like computes motion in the way that we're interested in. But it's actually not very tractable for trying to understand how motion gets computed on sort of a very fine-grained level. And so what we do in lab is instead of studying the human brain, we go after this little brain over here. So this is drawn roughly to size. So this is a fruit fly brain. It's about 1 million times smaller than a human brain, and it has a lot of advantages for research into in neuroscience. And so let me just walk you through a few of those. So the first is basically that it's just small. So you can actually get in there and have access to all of the neurons in the brain. And this is really not true with human brains or with even mouse brains, where if you want to get deep into the brain, you have to cut away like other layers. Here, you basically can look at any neuron in the intact brain. The other cool thing about fruit flies is that basically all the brains, like the brain of every fruit fly in lab, is basically the same. So there are neuron classes that you can identify by sort of their structure, and how they're connected doesn't change from fly to fly to fly. And this would, for instance, absolutely not be true of us, right? If we look in my brain versus one of your brains, like you'd be hard pressed to identify any neurons that were like equivalent. And there definitely would not be connected in sort of an identical way. Flies also have really nice, robust behaviors. So I'll show you some videos about that in a moment. And that makes it possible to really understand what the fly is doing and what it cares about. So behavior is this really powerful measurement for understanding how the brain works, because behavior is really the output of your brain. It's the final thing that your brain cares about. And then last, in fruit flies, there's because they've been studied for so long as a genetic model organism, there are a lot of genetic tools that you can use to both monitor neural activity, so measure from neurons, and also mess around with it, so turn neurons on or off. And so we'll talk a little bit about that today as well. So the processing properties are, I guess this is to reiterate, are shared across flies and mice and primates. So so even though the fly and the mouse and these two primates, these happen to be my primates, so these are my two daughters, the eyes all look very different here, but it turns out that a lot of the properties about how they detect and process visual signals look remarkably similar. And so that's one of the reasons that we can study the fly and learn about how human visual processing works. So are there any questions so far?
No, I read a little bit before your talk, and I will ask my questions later on. Thank you.
Okay, great. So I guess in the next, I'd like to just talk about what the problem is for detecting motion. So what you can do is imagine that you're a brain and you have an eye. And what your eye does is it detects the light intensity at different points in space, at different points in time. And then it just tells you what those intensities are, right? So you could imagine you get a bunch of signals from your eye that look like this. And then your goal is to take these signals and figure out a way to combine them in order to understand what outside stimulus or what outside in the world is generating these signals. And in particular, what you want to know about the world is whether it's moving, and if so, which direction it's moving and how fast it's moving. So that's kind of your goal as a problem. And it turns out this is not an easy problem. It's not the hardest problem that the brain solves, but it's not an easy problem. And just to give you a sense of how hard it is, I want to show you two movies. So in this first movie, we just have a scene. There's sort of noise added to it, but it's just so patently obvious that it's a scene moving to the right that this doesn't feel difficult, right? This is sort of such a low level perceptual process in you that you just don't even have to work hard to know that this is moving to the right. If what I do though is instead of giving you the light intensity, I give you a numerical value that reflects the light intensity. So now I'm just giving you numbers that tell you like how bright the scene is at different points in space. And when I do that, now all of a sudden you basically have no idea which direction this is moving in. And this now reflects like the fact that this is a difficult mathematical problem. One of the points here is that the reason this is so easy is that our brains have evolved to perform this this motion computation extremely efficiently and extremely well over millions of years of evolution. And so it feels easy, but that sort of masks the underlying difficulty of the problem. And to tell you about, or to sort of illustrate how difficult this problem is, I want to show you a visual illusion, right? So this, you may have seen this before. This is called the snake illusion. Edwin, let me answer your question in 1 second. so this is called the snake illusion. What you see are these sort of circles, and this is a static image that I'm sending. There is no motion in the image whatsoever, and yet as you look at this and sort of blink or move your eyes around and maybe look a little bit off to like the edge of your monitor, what you should see is that these circles are kind of slowly, they appear to slowly rotate every time you move your eye. And so this is your eye basically making a mistake, right? You're perceiving motion where there is none, and that's because this is a hard problem. And so what we want to do is understand how this motion illusion arises. Like why is this happening? And we're going to do this all in flies. And the first thing I'll tell you is that flies actually see a similar illusion, and I'll show you data for that later. Let me see if I can answer Edwin's question now. So the question, isn't that how people give images to machines? For example, facial recognition. So Edwin, do you think you can unmute yourself and ask the question? I'm not sure I quite understand from.
Oh, hi. Can you hear me well?
Yes.
Oh, so I've been really interested into machine learning, and I've seen some patterns like that, like boxes with like weights, like like numbers. And I saw that. I don't know if it's true or that's the mechanism that they use numbers to give pixels, I guess, density.
Yeah. Yeah. So I think this is exactly right. So when we, when your camera takes a picture, right, it represents the intensity of light at each point in the image by a number, right? And so that number then reflects the intensity, and that's how it's stored like on your hard disk. And so when you're doing any kind of computation with that image, you're manipulating those numbers at the different pixels in the image. So when we want to think about what a model might look like for motion detection, we want to think about how you can manipulate those numbers. When your brain is doing this, it has some set of like voltages or synaptic releases or that type of thing, and it wants to figure out how to manipulate those.
Okay, thank you.
So I just told you that flies actually see a similar illusion, and you might ask how we could possibly know that. And it turns out that flies have this very strong behavioral response to motion, so that when the world rotates around a fly in one direction, the flies actually turn to keep up with it. So in other words, if they see motion to the left, they will turn to the left. And so what we can do is set up in lab ways to measure fly behavior so that we can see what the fly perceives, what direction of motion the fly perceives. So what we do is we usually tether flies in place, and then we place them above these air suspended balls. And then what we can do is surround them by TV screens, basically, where we can show some visual stimulus, right? And then we can measure how the fly walks on this ball, right? So if the ball rotates backwards, the fly is walking forward. If the ball rotates to the left, the fly is turning to the right, and so forth. And it turns out, once you've built one of these, oh, my slide is not working here for some reason. Okay, here we go. Turns out once you built one of these, it's very straightforward to build many more of them. So we actually have 15 of these in lab where we can measure fly behavior in response to some visual stimulus. And then here I'm going to show you, in the interest of comparing flies with humans, this is Emilio who helped build this rig, and also Matt and Brian. And so Matt is showing that he can't really compete with flies in this sort of behavioral task. So I want to tell you a little bit about what's already known about fly visual circuits. There's this behavior that we can measure from the fly in terms of how it walks. And then there are a bunch of neurons that are generating this behavior. And so what does this look like in the fly eye? So at the very front end, we have this array of facets, which you've probably seen before if you've looked carefully at any insect. And these facets are focusing mechanisms for the fly visual system. And you might think that this is the type of view that the fly has of the world, that each of these little facets acts like a little camera. And so if someone were about to swat a fly, it would have like 100 copies of that image. But in fact, this isn't really how the fly eye works. Really, those facets are more like a pixel. So each of those facets act like a single pixel that the fly uses to compose an image of the world. And so it has this very low resolution view of the world. Each of those pixels represents about 5 degrees of visual space. And 5 degrees of space is roughly, if you stick your palm out at like arm's length, your palm occupies about 5 degrees of space. And so that's the size of a single pixel in the fly eye. And so that means that the fly has this very low resolution view of the world, right? So the fly is not able to read a book. It's not able to recognize a face. It's not doing a lot of the things that we think of as being really important in our vision. But it is able to do the things that are important to it. So what we can do is look at the fly eye and now go inside and go into the brain. So we're going to take a slice through the fly's head like this. And this is what that slice might look like. So these are the 2 eyes on the outside. These are the antenna at the front of the fly's head. And then this whole orange structure in here is the fly brain. And then this part over here is where the fly does all of its visual processing. So it's the part closest to the eye. And what we can do is look at this beautiful image that was made by coloring every neuron in the fly's brain a different color. And so this shows kind of what the sort of 50,000 or so neurons that are involved in visual processing here are doing, or what their anatomy looks like. And what we can do then is zoom in on some individual neurons that we know are related to motion processing. And so these are now examples of neurons that have very different, these are a bunch of different neuron types that have different characteristic morphologies. So you can see a cell body, for instance, and then neurites that target different layers and have sort of different looking axon terminals. And so these are the sorts of neurons that we're concerned with in lab. And I think the important point here is that these neurons are, there's not, even though I've drawn just one of them, like I've drawn just one of these red neurons, there is not just one in the entire brain. It turns out there's one behind every single facet. So you can think of each of these facets has sort of a column of neurons behind it, and then that is replicated behind every single facet in the fly's eye. So if you, for instance, light up one of these neurons with a fluorescent protein and then look at it, there will be many, many copies of it. So there's one, each of these corresponds to the neuron behind one of the facets in the fly eye. And you can see this beautiful hexagonal sort of lattice in this fluorescent image here lines up with the array of facets in the fly's eye. Now, for motion processing, there are 2 neurons that are known to be involved in detecting motion. So one is called T4, and it responds to light edges moving across dark backgrounds. The other one is called T5, and that responds to dark edges moving across light backgrounds. And so these 2 neuron types are, as far as we know, the neurons that detect all the motion in the fly eye. And it turns out at each point in space, there are 8 of them. So there are 2 that are sensitive. So there's one T4 that's sensitive to up and one T5 that's sensitive to upward motion, one of each that's sensitive to downward motion, one of each that's sensitive to leftward motion, and one of each that's sensitive to rightward motion. And that's just that's just behind a single facet, right? So if we now look at the array of facets, there are these 8 behind one of them, and then there are 8 behind every single one of these. So this sort of tiles all of visual space, and these neurons are known to be detecting motion that drives behavior in the fly. So what we want to do is figure out how all of this works. And we have a set of tools that we can use to dissect this circuit. So this is sort of now thinking about the logic of all of the experiments that we do in lab. So there's kind of a nice analogy that I like. So imagine that instead of a fly, you had a radio, and you're trying to figure out how this radio works. And what's cool about this thought experiment is instead of having one radio, you have an infinite supply of identical radios at your disposal, and you want to figure out how they work, right? And so how would you go about doing this? So the first thing you need to be able to do is identify the different wires and the different components in the radio, right? And so in flies, we actually have ways of targeting all of the different neurons in the fly brain. I said all, and that's definitely an exaggeration. We can target a lot of them, and more than in like other popular animals like mice. And so what this really does is this allows us to identify the different wires and components in the fly brain. And just to give you a sense of this was a beautiful paper that made 30 of these tools to target 30 different neurons. So just to, sorry, I should have said this here. So in purple here is basically an outline of the fly brain, and then in green, we're showing a particular neuron type. So this happens to be the neurons T4 and T5. And what this group did is they made a bunch of tools that allowed us to target a different class of neurons called LC4, LC6, and so on. And so each of these is an image of the anatomy of that different class of neurons. And then what's cool is because you can just send flies through the mail, we can send away and order any of these tools we like. And so you place an order and they arrive like a week later, and you can start doing experiments with them immediately. So this is like a wonderful set of tools for being able to understand how the fly brain works. One of the things we can do once we can identify a neuron genetically is we can express proteins of our choice in there. And one of the proteins we love is called Shibiri. And what Shibiri can do is it can actually shut down the neuron and turn it off. So you can think of this a little bit like having clippers, right? So what you can do is you can take your radio, and now you can like find a wire, and now you can just clip it, and you can see how that affects what the radio does, right? So maybe the radio no longer will change volume, it no longer will change station, maybe it just doesn't work at all. This is a way that you could imagine figuring out how each component contributes to the radio working, and that's how we're thinking about the fly brain. Yeah, and so this is very similar to optogenetics. This is kind of, so optogenetics is typically activating neurons, and this is silencing. So it's sort of the converse to most optogenetic experiments, although there are definitely optogenetic tools that silence as well. And those we would have a similar property for this. And the last thing we can do is use engineered proteins to actually watch what neurons are doing in real time. So for instance, there's a set of proteins that are green and will fluoresce under a microscope. And then when the neuron is active and when there's calcium in the neuron, they fluoresce even more brightly. And so you can think of this as having like an oscilloscope or some way to measure voltage in your radio, right? So you can measure what the components are doing. So this is sort of our suite of tools. We can identify different components. We can break them in various ways to see how they contribute to the function in the brain, and then we can measure what they're doing. I just wanted to show you an example of what this imaging looks like. So what we can do is place these flies under a microscope and then put them in front of a TV screen, right? And then we can show them some stimulus on this TV screen. And what I'm going to do is show you a movie of what our data looks like, recording in this brain structure. And the cool thing in this brain structure is that the neurons that are selectively responsive to up, down, left, and rightward motion are all separated out physically. So they're in different layers of this structure. So this data was taken by Emilio a few years ago now. And what I've done is I've outlined where the 4 different layers are here. And then I'm going to start this movie in a moment. This icon here tells you what the fly was seeing. So when it's a circle, the fly was seeing nothing on the screen. And then it'll be an arrow to the left, to the right, down or up. And what you should look for is the change in fluorescence here. This is roughly going to be real time, a little bit faster than real time, but you'll see when you see like a bright patch here, it means those neurons were active, right? And so you're watching the brain in action. So here we're going to see left first, and that lights up this orange layer here, followed by rightward motion, which lights up this blue layer, followed by downward motion, which lights up this yellow layer, and upward motion, which lights up the purple layer. So now what we want to ask is, can we figure out, using flies as a model system, why we perceive motion in some stationary patterns? So this was that snake illusion that we started the talk with. And so the rest of for the rest of the talk, I'll tell you about some of the progress we've made towards understanding how this sort of thing works. And the first thing we do is we want to change to a slightly simpler pattern. And so this is called the Fraser-Wilcox illusion, and it looks very similar to the snake illusion that I showed you earlier. It's a little bit less powerful in humans, but I think it's important to try to get you to see what this illusion is. So what I want you to do is, instead of looking directly at your screen and directly at it, I want you to look kind of off to one side, maybe at the wall behind the screen, or off to one side of the screen. And what you should see, and then pay attention to the pattern basically out of the corner of your eye, right? So don't look directly at it, but pay attention to what it looks like without looking at it. And what you should see is that this pattern appears to rotate very slowly clockwise. And what I'm going to do is sort of move it around a few times and put it in different places. And sometimes when it appears, it will have this, you'll see this effect more strongly. Sorry, I missed a couple questions here. Let me answer them right now before we go on. So one question is, why do these layers detect specific motion patterns? What makes T4 and T5 neurons in each layer different from the layer that's above or below it? So that's actually the main question, right? Is how do you make a neuron? So the layers contain different neurons, and so each layer reflects the activity of one set of neurons. So the set of neurons that are selected for up, down, left, or right. And then like one question is like, how do they become selective for up, down, left, or rightward motion? And that's exactly what we want to understand in lab. And I think we know something about that, but definitely not everything. But that's like a really good, excellent question. That is a very central question to what we want to know. Let me go back here. So Cielo, I guess, asked why did certain motions cause more excitation in the recordings? Was it because they were more obvious? Yeah, so it's hard to tell why. So that's a very good observation that some of those looked brighter than others. And I think it could be because of the way the screen was oriented. It could be because of the way this fluorophore was expressed in some populations of neurons versus others. It's a little hard to to gauge or to compare between populations of neurons. What we would typically do is look at within a population of neurons, so within a layer, like what does it respond most to, and that kind of thing. That's a great question. And then Carlos was asking, would diagonal motion activate a combination of two of these layers together? So that's exactly right. So what you can imagine is you have an upward sensitive neuron and a rightward sensitive neuron. And if you had diagonal motion up and to the right, it would activate both of those. And you can think of that as like a vector addition, right? So you can represent the direction of motion by these two vectors added together. And that's exactly how we think it works. And then Cielo again, related to Hector's question, how would they interact together? Yeah, so I think the way to think of this is, you know, if you were going to write down like this diagonal vector in terms of, you know, a horizontal and a vertical component, you would, that's exactly how the eye does this as well. So it decomposes that diagonal direction into its vertical component and its horizontal component. Okay, so back to the illusion. Actually, if people could put in the chat whether they can perceive this, that would be helpful to me. It's always a little hard to do this over Zoom. So if you do see it, oh, that's great. Okay, so I'm glad at least it's possible to see this over Zoom, and I'm going to move it around a little bit. It is very weak. So if you're just seeing it rotating slightly and you think you're maybe not seeing anything, that's still it. So when I move it to a new place, sometimes you'll see it like slightly more strongly when it arrives at the new place. And now I want to show you one version of this that you'll see very strongly. And if you haven't seen it, you'll see it this time. So now what I want you to do is sort of stare right at the center of the pattern. And I'm going to take it away. And when I take it away, you'll see motion to the left. Sorry, counterclockwise. So here it goes. And so when it disappeared, I hope you saw like a fairly strong percept of motion to the left. And so it turns out, so humans are not the only animals that see this. There are a bunch of vertebrates that also see this. So this is a cat video from YouTube, where this cat is looking now at the earlier version of this that we looked at. And you can see that it definitely appears to be pouncing and maybe seeing the same sorts of weird motion that we see as well. And it turns out fish and non-human primates also are susceptible to this illusion. So what we wanted to know is whether flies also saw this. So we started off with the illusion that I showed you just now. And what we wanted to do is see whether we could make flies turn using this illusion. So instead of sort of having it out in front of us and having the rotation be this way, what we wanted to do is kind of array this pattern around the fly. So like this, right? And so now what we know from the fly behavior is that if this fly perceived this as moving to the left in front of it, then it would turn to the left. And if it perceived it as moving to the right, then it would turn to the right. And so what this looks like in our experiments is that we sort of put this pattern with these dark to light transitions. We array that on the screens around the fly. And then what we want to do is see whether the fly turns in response to this. And that would tell us that the fly, or that would suggest that the fly is perceiving the same illusion that we are. And so we did that experiment, and this is what we found. So here I'm showing you what the orientation of these light to dark transitions looked like. And if we look at this, we will see motion to the right here. So this would give us the perceptive motion to the right. And now what I'm going to do is I'm plotting the turning of the fly in degrees per second. So this is basically how fast it's turning the ball underneath it. And we present the stimulus for 5 seconds in this gray box here. And so initially the flies are turning 0 on average, they're not turning. And then when we present this, this stimulus, the flies turn on average about 5 to 10 degrees per second for the duration of the stimulus. And then when we take it away, the flies turn in the opposite direction. And so this is pretty weak turning. This, 5 degrees per second means that it would take like over a minute to do a full rotation, right? So they're not turning fast. They're turning very slowly. but they're turning in the same direction as we would expect if they saw the motion that humans see. And when the stimulus is removed and the screen turns gray again, they turn in the opposite direction. And so this is like that counter rotation we saw when the stimulus disappeared. So we think this looks very similar to what humans see. Now, we wanted to know whether, so I told you about T4 and T5 neurons that are responsible for all the motion detection that we know of in the fly visual system. We wanted to know whether they were involved. So what we wanted to do is express this protein, Shibiri-TS, and this is a really sneaky protein. So basically, when the fly is developing and just living its life, it doesn't even know that this protein is there, as long as it's at room temperature. And what happens is when you heat up this protein and you bring it up to 30 degrees Celsius or so, it changes conformation. And when it changes conformation, it shuts down the neuron's synaptic transmission. So you can think of this a little bit like a Trojan horse, that the cells like that express this, so T4 and T5, basically don't even know that it's there until we start the experiment and we heat the flies up. And when we heat them up, then all of a sudden these neurons shut off. So what we're doing is essentially taking these neurons out of the circuit. We're clipping these neurons. And what we find is that, so here I'm plotting the shut down neurons are in red. So that's the behavior in the case of the neurons being shut off. And then the two gray curves are just genetic controls. And so what we see is that in the case of the genetic controls, we see the same behavior that I showed you earlier in wild type flies. But when we turn off T4 and T5, we basically eliminate all of the turning. And so this makes it clear that the turning is being generated by these motion detector neurons. And so what we wanted to do is figure out how these neurons actually respond to this stimulus, right? Because that'll tell us how this works. So we put our flies in a microscope and we surrounded them by these screens again. And now what we want to do is look at how T4 and T5 respond to the static stimulus at different points in along the stimulus. So remember, there are rightward T4 and T5s and leftward T4 and T5s at every point in space, right? So there's a whole array of these. And I'll just remind you what T4 and T5 respond to. So T4 responds to light edges moving across a dark background. T5 responds to dark edges moving across a light background. And then T4 and T5b respond to the mirror image, right? So this is now leftward moving light edges and leftward moving dark edges. So that's what these neurons respond to. And now there's a whole array of these, right? So there's one set of 4 of these looking at every point in visual space. And we want to understand what those like what that array of neurons is responding to. So let me show you how we display this, because this is complicated, right? There are neurons at each of these points in space. And so instead of plotting these as lines, we're going to plot these in color. So red means that the neuron is responding strongly, and white means it's not responding. And so now we have a T4 neuron and a T5 neuron, and it's this neuron looking at each point in space. And then when we show a white bar, and time is vertically here, right? So this is now a time trace. The neuron is not active. When the white bar shows up, it becomes active. And then when the white bar goes away, it goes back to baseline. So what this says is that T4 neurons are responding to these white bars. T5s are not. And if instead of white bars, we present black bars, now T4s are not responding and T5s are, right? And then we can see that, it doesn't have to be looking directly at the black bar. It can be offset by just a little bit and it will still respond, right? So that's kind of how to read this, is we're now looking at what the responses of these neurons look like to an entire image. So you can think of this as the array of neurons and how they're each element in that array is responding. Okay, so now to get into the good stuff, which is like how it actually responds to the stimulus. So here we have our T4As, and remember these are neurons that respond most strongly to light edges moving to the right. And you can see that they're responding here to the light part of this sawtooth stimulus, here and here. And now what we can do is we can present the sawtooth stimulus, the mirror image of it. So here the ramp from dark to light is to the right. And then over here, the ramp from dark to light is to the left. So these are just mirror images of each other. And you can see that the T4A neuron is responding strongly here, but it doesn't respond strongly to the mirror symmetric version of this. And this is going to be key. So it's responding most here and barely at all here. Now we can look at T5s, and this has the opposite pattern where it responds the most here and barely at all here. And we can look at T4B and T5B, and these are the mirror symmetric neurons. And so they're doing the opposite thing, right? So T4A responds the most here, B responds the most here because it's the mirror symmetric version. And so it responds most to the mirror symmetric stimulus. And same thing with T5B, responds most here compared to T5A is responding most there. So this is the pattern of responses that we found, and we can quantify that in kind of two ways. I think the most important way is this way that's down at the bottom that we'll look at it in a little bit more detail later, which is basically just a cartoon of what the neurons are doing. So it says, this pattern evokes responses in the rightward T4 and the leftward T5. And the mirror symmetric pattern evokes responses in the rightward T5 and the leftward T4. And so you can think about this, like what's actually happening here is it looks like the neurons are responding to these sort of sharp edges, right? The sharp transition from white to black or the sharp transition from black to white. So instead of this complicated stimulus with the gradient and the triangles and stuff, what we can do is instead just present a square wave, and we see the same thing. So here T4A is responding most to this light to dark transition. And you can see this is actually, so this is the light edge moving across a dark background. And so if instead of having this moving, you just make it stationary, that's now the pattern that T4A cares most about, right? So T4A is responding to the static pattern that looks like the stationary frame from the moving edge that it cares about. So T4 cares most about this, T5 cares most about this dark to light transition, and then T4 with the opposite orientation cares about light to dark in the other direction, which is the same edge here. And then T5 in this direction cares about the dark to light transition in this other direction. So that basically these neurons are responding to the static version of the edge stimulus they care most about. And that basically brings us to our model for how we think this works. So we have this array of T4s and T5s, right? And we think that this pattern activates this T4 and this T5. And since the fly turns in the direction of motion that it sees, the rightward T4s promote turning to the right, and the leftward T4s and T5s promote turning to the left. And so you can think of there being sort of a net behavior here, which is the difference between the rightward neurons and the leftward neurons. And now what we've seen is that the flies actually turn to the right in response to this. And humans, for instance, see motion to the right in response to this. And so what we know is that there's some kind of imbalance between these signals. And so maybe you can think of the T4s as being slightly impacting behavior a little bit more than the T5s. And so we think the behavior is this imbalance between these two sort of opposite signals, right? One of which, so T4A signals that there's motion to the right, and T5B signals that there's motion to the left, and then they're not quite canceling each other out, and that's resulting in this mild sense of motion that we have and that flies have. And so we can test this model. So the cool thing we can do, for instance, is we can turn off all of the T5 neurons, right? In which case what we expect is we expect there not to be some of this canceling signal. And so we expect an even stronger behavioral response. The other test we can do is we can turn off all of the T4s, right? And in that case, the T5s are the only ones on. They're going to cause behavior in the opposite direction, right? So now the other, like what would normally be the pink arrow is now gone. Only the blue arrow remains, and now we're seeing motion. We're seeing behavior in the opposite direction. So these are predictions that this model makes, and now we can test these. So the first thing we did is we turned off T5 using the same method, and that's this pink line. And the two gray lines are again genetic controls. And what we can see is that this pink line is not very large, not very much faster than the gray lines, but the flies are turning slightly faster in response to this stimulus. So that means by silencing T5, we've actually enhanced this illusion in the flies. And now we can also turn off T4 neurons. And what we see there is pretty remarkable, that we actually invert the illusory percept. So now when the flies see this, wild type flies turn to the right, but when we've silenced T4 neurons, they actually turn to the left. And so what this shows is that we were actually able to manipulate the strength and also even the sign of the illusory motion percept in the fly using these genetic tools that allow us to sort of break different parts of the system. So just briefly, like the ingredients for our model is that we needed these motion detectors to actually respond to static stimuli. And that was what we found in the fly. We needed this T4 and T5 to have sort of different influences on the behavior. And then we needed these signals to be subtracted somehow so that you would get this slight imbalance. Right. And so we think actually that we have a pretty good understanding of how this illusion works in flies. And then we've been doing some human experiments as well to see whether the mechanisms in humans look the same. And so far, it looks like the data supports a pretty similar looking model in humans. And so this is an example of how studying visual processing in flies can teach us about how our own brains work. So I'm just going to wrap up. So I first showed you that flies actually perceive these same illusions that we do, and that their motion detecting neurons are required for this, and that we've developed sort of a theory for why this happens that allows us to sort of predictably enhance or actually invert this illusory percept in flies. And last, I didn't have time to tell you about this, but we've done psychophysical experiments in humans that show that it looks like a pretty similar mechanism is likely at play in humans. So with that, I want to thank you for your attention, and also thank my lab who does all of the heavy lifting here, and especially the work I showed you today was done by Margarita and Ryosuke, two talented lab members. And so with that, I'm happy to take questions and discuss and talk to you guys about science.
Thank you, Damon, for a very, very interesting talk. I really learned a lot about how we see illusions, actually. Oh, Cielo has a question. She raised her hand. Cielo.
Yes, thank you, Dr. Clark, for sharing your time with us. It was a great presentation, and I wanted to ask you, as you may know, like in behavioral neuroscience, we have a challenging problem, and it's the fact that tracking models for tracking the behavior sometimes are not as sensitive to all the emotions the animals have. And I was asking if then these Drosophila models you researchers have been developing about how they perceive motion can be translated into biotechnology that would then result in tracking devices for behavioral neuroscience for more sensitive approaches like sniffing and tail moving and such.
Yeah, so I think this is a really good question, which is how should we measure behavior? And I think the way we do it is by sort of tethering these flies in place and then suspending them over this ball, right? And so what this means is that we're very sensitive to particular types of behaviors. So we're very sensitive to like walking and turning, but we don't know about all the other behaviors that the fly is exhibiting, right? So it could be moving its wings and we would never know. It could be moving its antenna and we wouldn't know. It could be grooming, like moving its legs but not moving the ball, and we don't know about that sort of thing. And so I think what you're talking about are there are a lot of like video capture methods and machine learning methods related to analyzing videos that are becoming incredibly powerful ways to sort of automatically quantify behavior. And I think those are super exciting. I think there was a paper published today actually on how you could do this with mice and rats, and how you could see like the effects of particular drugs, just like caffeine. You could like see the effect of the caffeine in the behavioral repertoire of the animal. And so I think those sorts of behavioral analyses are going to be really powerful for drug discovery and that kind of thing. You can, I guess a while ago, there was a beautiful paper that used behavior in zebrafish larvae to classify unknown drugs and sort of relate them to known drugs. And so what you do is you put a bunch of fish in little wells, monitor their behavior in some way, and then you dump different drugs into the different wells. And you have a bunch of known drugs where you know what they do and how they act, and you have a bunch of unknown drugs, and then you just sort of cluster. And so you say, like, the fish with unknown drug A, did it look, what other drug did it look closest to? And it turned out to do a really good job at helping classify how these drugs were working, where they were acting, and that kind of thing. So I think it's exciting to be able to measure behavior like this with this sort of precision.
Thank you, but I was referring to the fact if we can replicate the visual perception sensitivity of the Drosophila
I
to
see.
develop biotechnology to then measure in
Okay.
other experiments.
Sorry, I didn't get your question. So
No, it's okay.
I guess the so flies have eyes that are incredibly specialized for what the fly wants to do. And I'm not sure that what the fly wants to do is what we want to do when we, for instance, are developing like new ways of processing data and that kind of thing. I think the hope is that the algorithms and sorts of mechanisms that we find in the fly can help us come up with maybe come up with ways to process images that are maybe more robust to noise and that kind of thing. So I think I'm certainly hopeful that these things will work out. There's always a question of whether like the way biology solves things is the way that we should solve them, right? So right, like birds are really good at flying, but when you want to make an airplane, you don't necessarily want to deal with like actuated wings. You just want to hook a jet engine to a big metal plane, right? And so I think it's I think we can learn from biology like what sorts of solutions are possible, and then if those solutions that the biologists come up with are really what we want, then I think I'm very excited to see whether they have applications.
Great, thank you. Wonderful talk.
Thank you. Any other questions before we go to the...
Yes, I had a quick question, Dr. Clark. It refers to the imbalance that you mentioned in terms of your model.
Mm-hmm.
I just wanted to make sure if I was clear. When you mentioned that imbalance in, I guess, neural activity between the T4s and the T5s, is that pattern specific? Like, I don't know if I'm making myself clear.
Yeah. So I think your question is this sort of a general property that T4s are always like stronger?
Exactly.
Yeah. And I think we believe the answer is yes. So we think that T4s generally exhibit a sort of stronger influence on behavior than T5, like for equivalent stimuli, the way that we're defining equivalent, right? So I think a lot of this depends. So if you think so the way we would measure this would be we'd have a gray screen, and then like we'd have a white edge come across it, and we'd see how fast the fly turns. And now you have a dark edge come across it. You see how fast the fly turns. And the fly will turn slightly faster to the light edge. But whether those are equivalent is not totally clear, right? So I guess like given if you think those are equivalent, then T4 is responding more, is having a larger influence on behavior, and the equivalent is kind of how we built our stimulus, right, where we went from black up to white. So our stimulus kind of has this built into it.
Okay. And do you guys have any hypothesis onto why that might be the case?
Yeah, we do. That's a good question. So we think that this is because fundamentally, if you look at the world around us, it's not actually light dark symmetric. So we often think of like light and dark as, you know, good and like two sides of the same coin, right? But it's actually the world consists of mostly dark. Most regions in the world are darker than average, with a couple very, very bright spots. So that's what most of the world looks like. And so it turns out, if you design an artificial motion detector and you ask it to perform well on a database of like natural images, it will also develop the same imbalance between light and dark that we see in the fly. So we really do think that this is an evolved property of the visual system.
Yeah, and it makes sense. Thank you so much for your answer.
Any other questions? So, Damon, the flies only see black and white. Are they Daltonian?
Yeah, you would think that from my presentation, but that is you're absolutely right. So flies actually have 5 photoreceptor types. So they can see, in principle, more colors than we can. So we have 3 roughly that we use during daylight hours, and so flies in principle they can definitely see things like UV that we cannot. They can also see other weird dimensions in the light that we don't. So they can see polarization of light that we don't see. And so their view of the world is probably quite different from ours.
Interesting. Learning. Anything, other questions? Jóvenes? Everyone? Okay. So, Damon and Dr. Greer, if they want to move to the other side, you know,
Yeah.
as you wish, as you wish. It's
Perfect.
up to you. You have plenty.
So Charlie, should I start with this like short presentation from Giovanna, and then you can
Sure. That's fine.
We'll trade from there. Okay, let me. Okay, so this is a presentation that is not mine. You'll notice that I am neither Giovanna nor Rochelle, but they were kind enough to give me this presentation, and this is sort of specifically related to Yale and its long-term ongoing relationship with the University of Puerto Rico, and also Puerto Rico more generally. So I'll try to do my best in presenting this and do it justice. So I guess this sort of starts with talking a little bit about Yale's relationship with the University of Puerto Rico, and then we'll talk a bit about some programs at Yale aimed at getting undergraduates into lab at Yale, and then the PhD programs at Yale. And I think that's where, so Charlie is the director of the Neuroscience PhD program at Yale, and so he'll kind of take over. And Charlie, you should of course chime in throughout as I give this. I think yeah. So we'll sort of hand off in probably a few minutes here. So Yale. This is sort of a beautiful picture of Yale's main campus. It's one of the oldest universities in the world, and probably the best thing about Yale is getting to have like wonderful colleagues. Both that's what I like best at Yale, and having wonderful students. And the students, I think, enjoy having like really good, talented other students to interact with. So I think that's really the pitch for a place like Yale. Down here, this best pizza in the U.S. is no joke. So there's a lot of friendly competition between several really amazing pizzerias. So don't ignore that when you come here. So Yale has a long history, or long in some dimensions. So it's had with University of Puerto Rico. So it's had sort of a collaboration with the University of Puerto Rico since 2015 and runs a joint MD PhD program with the university. Yale scientists have been sort of heavily involved in sending relief to Puerto Rico after the hurricanes in 2017. Yale also has a very close relationship with this Ciencia Puerto Rico that I think is that my colleague Danielle Cullen-Ramos is heavily involved in. So this is sort of has a lot of connections with University of Puerto Rico through that. And then the Latinx Center on campus was founded in the 70s by a group of Puerto Rican students at Yale. So there's sort of this long history there. I guess more recently, Yale was spent a lot of energy helping get COVID testing to Puerto Rico pretty early in the pandemic. And this is through a group of Puerto Rican scientists at Yale. There's a pretty thriving Puerto Rican community at Yale, in New Haven, and in Connecticut more broadly. And then this is a photo. So this is Danielle Cullen-Ramos, and then this is Residente, who came and visited the Yale campus and came to a bunch of labs, including mine, to sort of learn about neuroscience for one of the albums he was working on. So I guess what I wanted to do is start with undergraduate programs. So these are programs that are aimed at current undergraduates, and there are a bunch of them at Yale. And I think the sort of regrettable thing this summer is that it looks like because of the pandemic, these are not running. I'll send these slides. I'll send these slides as a PDF to Professor Maldonado, and so she can distribute these around. So this will have like the websites and stuff in them to look at. The ones I've looked at so far look like they're not taking undergraduates this summer. So I think this is an unfortunate effect of the pandemic, but you know, I suspect they'll take twice as many students next summer, or at least this is my hope. So I think that there are things you should look at because they're really very nice programs to get students into the labs at Yale. They pay a stipend plus travel expenses. And you end up living in New Haven for the summer. There's a lot of sort of extracurricular activities designed for sort of professional development and that kind of thing that are associated with these. So there's this Biomed Amgen program. There are a bunch of other ones. So I'm most closely associated with this physics, engineering, and biology one, but there are a whole slew of these. And so again, I'll send this out. So don't worry about writing down all of these names. There are also these diversity preview days. These will actually take you to campus if you're interested in Yale's PhD program. So this is definitely something to look at. So I guess this leads into the BBS PhD program. And so here's where Charlie will start to chime in. So basically, this is an aerial picture of Yale's campus. This is kind of the old campus in here. And then I'm up here on Science Hill. Charlie looks like he might be home now, but if he were in his office, he'd be over here at the medical school. And it's about a 15-20 minute walk between these two. And then there's a whole other campus called West Campus that has a bunch of institutes and a lot of great research at it. That's another 15-minute drive away. So that's a little bit further from the main campus. But this is how this axis, I guess this triangle here is roughly where all of the biomedical research happens on campus. So there are 380 faculty in sort of biosciences across these three campuses. And I guess one important thing is that a little over 100 of them are associated with neuroscience in some way. So there's sort of a massive footprint of neuroscientists across the campus. There are 8 tracks that you can apply to become a PhD student in biological sciences at Yale. So there's sort of this biochemistry, This is, I think, a little out of date. So this is biochemistry and biophysics and quantitative biology. There's a computational biology program, immunology, microbiology, molecular cell and genetics and development, molecular medicine, pharmacology, neuroscience, which Charlie will tell us about, and then plant molecular biology. So you can arrive at Yale through any of these tracks. And one really important point is that once you're at Yale, you can work with anyone. So you're not restricted to, if you apply in immunology, but you come here and then decide your real interest is plant genetics, you can just go do that, right? So that's a great property. So basically all of the faculty are open to you once you're here. So applying, Charlie, so why don't I just finish this bit and then I'm gonna hand off. Does that make sense?
Sure, that's fine.
So when you apply, there's a statement of purpose and a diversity statement, 3 letters from ideally academics who know you well. There's no GRE required. And it's actually it's not only not required, it's not even reviewed. So if you do submit a GRE, like it won't even be looked at, depending on, I think that's now the policy across all the tracks.
Yep, it is.
The deadlines are December 1st. So this is unfortunate. But this gives you 11 months to notice for when the next application cycle will be. And just a sense of like what, Charlie has served on many admissions committees and I've served on a few since I arrived here. And so I think probably the most overwhelming, the most important thing is research experience, like that more than grades. That's what really matters, and also to an extent, like to how did you take advantage to the opportunities you had, right? So I think there's wide recognition in the admissions committees that not every undergraduate has the same opportunities, but how are you taking advantage of what's available to you, right? So I think that's what we really value.
Exactly.
You probably have much more to add there, Charlie. You should chime in.
I was just agreeing with you completely. We take into account the resources that students have available and judge their accomplishments accordingly. It's not fair to judge someone coming from a small rural school in Colorado, for example, with someone that's had the opportunity to be at Harvard or Yale. So we're taking really a holistic approach to studying the applications. Grades are fine, but they're certainly not the most important. Research experience is right at the top. Letters of recommendation. These are the people that know you the best. So we depend upon them to tell us who you are. And I always tell my own students, when you approach someone for a letter of recommendation, tell them that you're going for a very competitive program and that you need a strong letter. And if they're not prepared to do that, that's fine, that you can move on to someone else. But don't be intimidated about asking them. And then the personal statement, again, we just would like to know who you are. And it's also very important. Why do you want to come to Yale? Sometimes people will tell us in their application, I want to come to Yale because it's a famous school. That's not good enough. We want to know why you want to pursue the sciences, who you might want to work with, and how that fits in with your future goals. And I'll tell you in more detail about the neuroscience program, if I can go ahead and share my screen.
Sure. Can I say one thing just about graduate school period? So I think this, so PhD programs I think are the most unrecognized, valuable educational opportunity available. So like not only do you not have to pay your tuition, you get paid to be a graduate student for the entirety of your time at Yale. You get paid a stipend of $37,000 a year, and Yale pays your tuition. You get health insurance, right? This is and moving expenses, right? This is a real like wonderful opportunity. So I think I wanted to chime in with that. And here, let me let you share your screen now, Charlie. Here.
Can you see that okay?
Yes, we see it. Yes.
Okay. So as Damon said, the organization of the graduate program in the sciences is under an umbrella program called the Yale BBS, Biological and Biomedical Sciences. We have 8 different tracks, as he mentioned. The INP, the Interdepartmental Neuroscience Program, is, in my opinion, the best one, of course. And the reason it's interdepartmental is because neuroscience transcends so many different departments. As you just heard from Damon's talk, from biology, physics, neuroscience, and so on. So we have 23 different departments that are fully engaged in the interdepartmental neuroscience graduate program. Damon already talked about all 3 campuses, the number of faculty, the different tracks, and so on. As you look at the different tracks and think about applying to one, and you do have to specify one on your graduate application, that's usually done in the context of looking at the faculty that are within each of those tracks and the kinds of people you think you might want to work with. This is not a hard and fast rule. We're not going to hold you to it. If you want to say in your application that you want to work with me, but you get here and discover that Damon's work or someone else's work is much more interesting, you can make that transition without a problem whatsoever. It's a diverse environment, and we want students from a variety of backgrounds, all backgrounds, to apply. Right now, we have approximately 72% are US permanent residents, 20% from URMs, 28% international, and a relatively even split between women and men in the program. Student support, Damon gave you the figures for the current year. For the coming year, it's going up to $3,850 with $45,700 in tuition also covered. And then health insurance is $2,450. So collectively, coming out with an undergraduate degree, that's not a bad starting salary. And certainly with the $38,050 in New Haven, you can live quite well, and you can certainly afford to sample all those wonderful pizza places that Damon mentioned. I have to add, I always mention the pizza during recruitments because it is fabulous. New Haven was one of the first places that introduced pizza in the US. And these places that are so competitive, their ovens were built in the 20s and the 30s, and they continue to use the same ovens with wood firing, and it's just spectacular. Advisory support starts right away. In fact, it starts before you get here, talking with the directors like myself, as well as the staff. We have a program manager in the INP, Carol Russo. We have the BBS director to talk to. Each of the tracks has a minority affairs director and staff, the graduate school deans and staff, and professional development that's available exclusively to graduate students, talking about career goals and the way to pursue those goals. The deadline for applications for this coming year will be December 1st, 2021, so think seriously about it. The things that are most important to us: statement of purpose, which includes your experience in research, your perspective on diversity, letters of recommendation, and as it says here, the GREs are not accepted. In fact, they'll be redacted if you have them sent to us. We think that they're not an adequate, they're not an appropriate, and they're not an accurate measure of people's abilities and opportunity for advancement. So the Interdepartmental Neuroscience Program, it's university-wide doctoral program. The degree is conferred by the Interdepartmental Neuroscience Graduate Program. As of today, we have 130 faculty members from 23 different departments and programs throughout the Faculty of Arts and Sciences, as well as the medical students, and approximately 115 students pursuing their PhD. It is an extremely broadly based program. I mean, you heard about Damon's work today, going more fundamental or more basic. We have folks that are working on cryo-electron microscopy to understand the structure of ion channels. And if we go the other direction, we have folks that are using fMRI to understand connectivity in the human brain and how that changes with experience. And a number of people that are interested in neurological diseases, schizophrenia or traumatic injuries, and how those may be addressed in the context of neuroscience. So a very broad program with a variety of opportunities for folks. We've had people come in with the expectation that they're going to be doing fMRI in humans, and they end up working on Drosophila, simply because they didn't realize the opportunities that might have been available there. So when you get here, we want you to explore and find out what the different opportunities may be. Admissions, there's a broad-ranging expertise of the faculty, and those faculty are very anxious to recruit new new students with experience throughout the neuroscience and related fields. We look at all your credentials, your volunteer activity, your service to the university, to the community, your research activity, and your interest in your statement of purpose and your prior research. When you write this statement of purpose, please have several people read it and ask them to get out their red pens when they do it. I can assure you that when Damon and I write a grant application or send a paper in for peer review, we have people read through it and ask them to criticize it heavily for us so that we can have the best statement possible. And I encourage you to do the same. Strong and informative letters. Letters of recommendation from your chemistry professor that says that you came to class every day and sat in the front row and you did very well in the course and you got a good grade. That's not useful to us. We want people that know you and understand you, beginning in particular with the people that you've done research with. Lab rotations. We think it's essential that you get in the lab beginning on day one. We actually start with a boot camp that Damon helps run for 2 days on computational approaches in the nervous system. And then we want you in labs doing research. We want you generating new ideas and knowledge. You've been sitting on your butts in classrooms for a long time now. Now it's time to go in the lab, generate new knowledge, push on that barrier of understanding of the neuroscience, push on it hard so that we can really advance. And that's the commitment you're making when you go into the neurosciences. So during your first year here, you have 2 semester-long rotations are completed. A 3rd rotation may be considered during the summer, after which you are expected to affiliate with a thesis lab. If after doing 3 rotations, you just can't make a commitment yet, you need 1 more, that's fine. I'll approve you to do another one in the fall semester. But then we want you to make a commitment. We love having you here, but we don't want to have you here forever. We want you to move forward, develop a successful thesis, defend that thesis, and then go out into the community and tell them what a great education and what the great opportunities that you had at Yale were. Coursework, as I said, we think getting in the lab and generating new knowledge is the most important. So we keep coursework to a minimum. In the fall term, there's a Principles of Neuroscience, which is a seminar, broad review of neuroscience, and then also the foundations of cellular and molecular neuroscience. Spring term, we have Foundations of System Neuroscience, which is paired with a very short course on comparative neuroanatomy and ethics. You need to consider ethics in the context of appropriate behavior when we're designing experiments. What are the ethical implications of using neuroscience in the courtroom for law? How do we deal with conflicts of interest in the lab and so on? During year 2, there's a major milestone. The first part of the year, you need to pursue a course in quantitative or quantitative methods or analyses. We have several courses that are neurospecific for that, but we can help you select the course that's best for you based upon your prior experience. And then after that, you'll also be needing to complete 2 elective courses, your choice prior to the end of the 3rd year. They don't have to be neuroscience courses. We have one of our 2nd-year students this year taking a course in the philosophy department on the essence of experimental design. So we're open to really great flexibility on the electives. But the big milestone in year 2 is the qualifying exam. You identify 4 members of the faculty, not your thesis advisor, whose work interests you and whose work may influence the way you think. And with each of them, you read a series of 10 to 12 papers, different papers with each of them reflecting their expertise. You meet with them on a regular basis over a period of anywhere from 6 to 8 weeks, depending upon your scheduling. And on a weekly basis, you go through in excruciating detail 1 or 2 papers, understanding the whys, the wheres, and the what comes next. I think it's a very privileged opportunity. You get the opportunity to sit down and have a one-on-one tutorial with some of the greatest minds in neuroscience that are available in the US right now. You can learn how they think, how do they approach a problem, how do they resolve a problem, what do they think is important, and where do they think their field is going. During year 3, if you have any remaining electives, you take those. You also need to consider completing the teaching requirement. You need to teach for 2 terms as a teaching assistant. But the major milestone in year 3 is the thesis prospectus, and you advance to candidacy as a consequence. That has your thesis advisor as one member of the committee, And 3, or if you really want to, even more members of the thesis committee as well. We encourage you to keep it to 3 outside members besides in addition to your thesis advisor, just in terms of the convenience or the realistic opportunity to get everybody in the same room twice a year to hear what you've been doing and how your progress has been. Right now, our time to degree is approximately 5.4 years. NIH would like us to get down to 5 years, and we'd like to do that as well. Again, in the context of, we love having you here, but we want you to continue to pursue your career. We don't want you to stay with us for too long. Teaching and seminars, I already mentioned 2 semesters as teaching fellows in courses of their choice. Often that isn't, in fact, a neuroscience course, but if you choose to work with John Carlson, for example, who's one of Damon's colleagues, who teaches introductory biology, you can do that. Or maybe it's a math course or a quantitative course, or our Principles of Neuroscience course. It's going to be your choice. We have a wonderful seminar series. The Interdepartmental Neuroscience Program works with all the different departments around the institution to organize and bring in seminar speakers on a regular basis, usually Mondays at noon. The INP specifically supports the recruitment of seminar speakers, and those that are recruited by the INP are at the initiation of the students. So the students identify and select the speakers that have come on behalf of the INP, and they are responsible for hosting them, introducing them. They take them to dinner in the evening when we're able to go to dinner again without any faculty members. So they really have the opportunity to get a one-on-one and with faculty from other universities working in other problems, and learn about how people think, outside of Yale. Don't just depend upon us. We have student faculty lunches to pursue topics of common interest. Sometimes the students have asked us to talk about the process of grant applications. Other times we've had 2 faculty, one from a smaller university that's also in Connecticut, along with one of our faculty from Yale, and they've compared and contrasted what is it like being a scientist and a faculty member at a large high-power university like Yale versus a smaller one. So the topics are again identified by y'all, and you also determine who will come and interact and spend time with you. Mentoring and career counseling, extremely important. Year 1, there's an advisory committee of the INP faculty that's assigned to each student at the time of matriculation. And importantly, our students have now recognized the important, not recognized, they're extending even further the importance of advisory committees. So every student that comes in is assigned to a senior student who's their mentor and guide through the program. They can meet together as a group, or they can meet one-on-one. But the important part is this more senior student that has been through all these processes, the qualifying exam, the thesis prospectus, teaching, lab rotations. They're there to support you, to guide you, and give you better insight into the program. Year 2, it's the 4 members of the faculty that are selected by you to join their qualifying exam. And finally, in year 3, it's your lab PI and your recruitment of faculty to join that committee. In addition, I'm always there. And Damon is always there. We're all always there. And our doors are open. Even though we may or may not be a formal part of your advisory committee at year 1, year 2, or year 3, we're always there. The door is open and happy to explore with you your achievements, your excitement, your concerns, whatever it may be. We want you to be successful and happy. Our objective, our goal is that you have a successful career and provide you with the tools that you can go forward from here and make a big difference. This is from our retreat from 2019. That's me right there in the middle. I didn't find Damon, but you can see there are a number of faculty. We do this on an annual basis, and these are some of the broad areas of research expertise that are represented in the neuroscience track. So it's a great track. It's an exciting opportunity. 130 faculty, as I mentioned, with extremely broad interests, and we are extremely interested in recruiting people to come and join us at Yale. In 2019, so this was the numbers from a year and a half ago, we have 109, now we're up to 115. We have 59 women and 50 male at the time. 86 are straight PhD, 23 are MD PhD, 35 were international, and 74 were the US citizens or permanent residents. Academia, 54% of our graduates stay in academia. 27% going to industry, consulting, pharmaceutical, biotech, 7% going to teaching. We had a student several years ago that did some fabulous work on small GTPases that regulate the formation of dendritic spines. He had an interruption or he had a delay rather, in when his postdoc was going to start, but he wanted to move to New York because that's where his significant other was living. And in order to accommodate that, he got a job teaching biology in a very exclusive school in New York. He loves it, and he's still there. He never left. He didn't realize what teaching and expiring in high school students could be like, so he stayed. 4% go into higher ed administration, deans and such, and about 3% go into the government or other science-related areas. And as I said, time to degree is 5.4 years. This says it all. Please apply. We want to have the opportunity to meet you. We want to have the opportunity to talk to you, and we're looking forward to hearing from you. You can contact me down here at the bottom. Carol Russo is our program manager. She's always prepared to talk with you and would look forward to hearing from you. I will tell you that it's not easy. It's not easy being in graduate school. It's demanding, requires sacrifice in terms of time, and maybe you don't get to go play as much as you used to. And the application process is also tough. The number of offers that we can make is very limited, but we're very much looking forward to seeing your applications and making it possible for you to come and interview with us and perhaps join us to pursue your PhD.
One other thing I'm not sure Charlie mentioned is when you go to the website, it's going to tell you that you need to pay a fee to apply.
Oh yeah,
which is ridiculous. So do not pay that fee. Click on the link to apply for a fee waiver, and that will be granted to you. So do not pay money to apply to this program. You should get that fee waiver.
Yeah, and if you're applying to other programs, and you should apply to other programs, because otherwise you don't have the opportunity to find out what they may offer. Those programs will also cancel that application fee.
Yeah, so do that everywhere.
So Hector's been to visit us before, I think at least once or twice during earlier virtual events. He may have some questions, I'm not sure, but I'd be thrilled to answer questions that any of you might have.
Any questions? Well, okay. Well, this is.
I don't have a question per se, but I just wanted to say that Dr. Greer mentioned that the whole support and mentorship process starts even before you get there. And I just want to echo that that's completely true. Like throughout this entire interview process, I'm currently interviewing for the neuroscience track at Yale. And throughout the entire process, I felt that the mentorship and the support from the diversity initiative, the diversity initiative collective at Yale, and Ms. Russo and everyone over there has been outstanding. And I feel that it's really helped me feel confident in my decision to apply to Yale. So yeah, I just want to echo what Dr. Greer said and just let you all know that it really is true. Thanks,
Hector. It's great to hear.
Yeah, you have an advocate here. A very strong one. Sounds like you will be seeing him soon.
Well, we're talking next week during the first phase of our recruitment. Yes.
Wonderful.
Looking forward to it.
Well, I'm not biased, but Hector is wonderful. So you have a wonderful candidate. So
that's what we're thinking.
Yes, good to hear. Well, I guess if there are no more questions. Oh, Paola has a question. She's also wonderful.
Okay.
As the summer program deadline passed, but students with fellowship can have opportunity to have summer research experience. Well, I think Damon answered that you guys are not going to have summer this year. Can you?
Yeah, this was the notice that went out like a week ago that was very disappointing, was that I think there will be no undergraduates on campus this summer from who are not Yale students. So I think this is very unfortunate.
It is. We're looking forward to next year, but I will add that I have one student this year that's doing a remote lab visit with me. We talk on a regular basis. I'm interested in early development and synapses. And he and I talk. He joins our lab meetings. He's responsible for presenting papers in the lab meetings as he would be if he were here in person. So if you recognize someone at Yale or any institution that you would be interested in working with and you want to get to know them a little bit better, send them an email. Ask them if you can have a virtual rotation with them.
And I think the other so I looked on these websites. The websites haven't actually canceled the program yet, and I know what they did last summer is that they went to these all remote research summer programs, and those work, right? Like you can get a lot done, especially if there's data that the lab has collected and that you're interested in analyzing, if you're writing like a review or researching like a new topic for lab, and you can talk to a graduate student or a postdoc like every couple days about like what you're learning and what to read next. Like these things can be very, very valuable. So I don't think that just because the in-person stuff isn't happening that nothing is possible. And I don't know how the programs all learned about this last week. So I think none of them, to my knowledge, have canceled these. It's just that it's not going to be in person. So they're going to look different from what we would normally describe. So it might be worth, as I said, I'll send Dr. Maldonado that slide deck. So that has all the links for the different versions of these programs at Yale, and then you can do a little bit. I would give them like a week or two to figure out like how they're gonna handle this change, and then see what they have to offer.
Great, thank you for the information. Okay, I think we're done with questions. I think so. Well,
Great. And so yeah, thank you guys so much for spending time with us today. And if you do have any other questions that you didn't think of just now, you should email me or Charlie. I think our emails are easily findable online if you didn't note them down in the presentations. So, and I'd be happy to point you towards, if I can't answer it, I'll point you towards someone who can.
Yeah, I would echo that. It was a pleasure to have a chance to talk with you. And certainly my very best wishes as you pursue your goals in the neuroscience. And I hope we perhaps have the opportunity to help you meet those goals.
Wonderful. Thank you so much, both of you, for being here. And I really appreciate that you are part of, we end our series with Yale University. So our BP Endure ends with a golden end, I think. It's wonderful. And I really appreciate you being here. And to the students, take notice that Yale is recruiting, and so please, we will have all the information for you either our website or through my email, or you can contact directly Dr. Clark or Dr. Greer. And most certainly, we have several of our students that have come from UPR Rio Piedras and now are being successful graduate students at Yale, including our very own dear Daniel Colon Ramos.
Yep.
So thank you so much. Have a wonderful rest of the week, and have a great semester.
Great.
And
Thank you.
Be safe and healthy. Thank you so
Thanks
much.
again. You too.
Thank you. Bye-bye.
Bye.